Welcome everyone, and thank you for joining us today for our webinar on storage and handling of reagents on microfluidic cartridges. This presentation is the first in a planned lecture series focused on innovations in the design and manufacturing of microfluidic devices for diagnostics and analytical applications. We're starting the series with a core topic, microfluidic point-of-care devices. Today's session is recorded, and we hope it will serve as a useful reference for both you and your colleagues. We welcome your feedback and questions at any time. Please feel free to reach out to us by email at thinx-sales@idexcorp.com. You'll also see this contact information again at the end of the presentation. With that, let's dive in. One defining feature of point-of-care microfluidic systems is their ease of use outside a traditional laboratory setting. To achieve this level of simplicity, all reagents required for an assay must be stored directly on the consumable cartridge. This requirement introduces one of the key challenges in system design: long-term reagent storage and stabilization. Despite its importance, reagent storage is often underestimated during development, even though it directly affects device reliability, usability, and large-scale manufacturability. Serial production, in particular, is a theme we'll return to throughout today's discussion. The goal of this presentation is to review the challenges when developing point-of-care devices, review the overall development process and place this within the context of choosing the right reagent storage concept for a particular application, provide an overview of established on-cartridge reagent storage concepts, and introduce newer, more advanced approaches beyond common standards, and discuss their advantages and limitations. We hope today's session offers useful insights and perhaps even sparks new ideas or collaborations. Thank you again for joining us. Let's get started. When developing a new point-of-care cartridge, the assay itself is the primary design driver. This includes the assay workflow, reagent requirements, performance specifications such as sensitivity and reproducibility, and any regulatory or usability constraints. Every development must address a few key parameters. Minimize user interaction. Reducing user steps lowers the risk of errors and makes the device usable with little or no training. From a reagent perspective, this means all reagents should be stored directly on the cartridge. Enable efficient fluid management. The storage concept must support reliable reagent release, accurate metering, proper mixing, and, where necessary, reconstitution. Ensure reagent stability. This includes compatibility with manufacturing environments, storage conditions, and material interactions. Support large-scale manufacturing. The storage solution must be scalable, robust, and cost-effective for high-volume production. When we revisit these requirements, it becomes clear that the choice of reagent storage concept should be made as early as possible in the development process. Let's briefly zoom out and look at how a typical development project is structured. Most projects follow a classic phase gate process, where progress is reviewed at defined milestones and decisions are made on how to proceed. At the start, all cartridge requirements are analyzed and an initial concept is created. During the feasibility phase, critical functions are evaluated, often using test modules rather than a fully integrated cartridge. The integrated cartridge typically emerges during the design phase, where early prototypes are developed and design changes are still possible. Once the design is frozen, the project moves into the production phases. Ideally, the reagent storage concept is selected during the concept phase, when functional, manufacturing, and cost considerations can all be addressed together. It can be tempting to begin with solutions that are easy to implement early on. While these may work well for proof of concept testing, they often fail to meet the demands of scalability and high volume production. This can lead to major redesigns later in the project, resulting in delays and increased costs. In our projects, we focus on reagent integration approaches that are both well proven and highly adaptable. These scalable, standardized concepts can support development all the way from early feasibility work right through to final production. This slide illustrates that approach using dry reagent storage as an example. Here, a single concept, the reagent plug, which we'll discuss in more detail later, is selected early. While the plug can be adapted to different assays and requirements, the fundamental concept remains the same, from initial design through feasibility testing and into production. By relying on existing standards and test platforms, unnecessary development loops can be avoided, ultimately reducing time, cost, and risk when developing a new cartridge. With that foundation in place, let's move on to specific storage concepts, starting with liquid reagents. Liquids are typically stored in separately prepared containers that are later assembled into the cartridge, or in integrated liquid tanks that form part of the cartridge backbone. We'll begin with separately prepared containers. Several options, including blister packs, stick packs, or glass ampules, have been proposed. Most commonly, however, blister packs are used. A standard blister pack consists of a cold-formed aluminum film that creates the blister dome, combined with a second aluminum layer that forms the base. The blister is usually attached to the cartridge using a double-sided adhesive tape. To release the liquid, the cartridge either incorporates a piercing mechanism or uses a blister designed with some sort of frangible seal. In both cases, the blister is compressed with a plunger to dispense the reagent. This approach is reliable and flexible in terms of storage volume, but it does have some drawbacks. Blisters are typically filled in an open state and sealed in a separate thermal process. This can impact reagent stability and often results in residual air inside the blister, making liquid displacement less consistent. On the next slide, we'll look at a more advanced blister solution that addresses many of these limitations. The thinXXS blister pack technology significantly reduces material usage and simplifies processing. In this approach, a cold-formed aluminum foil blister is sealed directly onto the microfluidic chip, creating an integrated wet reagent storage solution. Reagents are filled afterward through dedicated fill ports on the cartridge. This enables filling directly at the customer's site and supports a true platform strategy, where a generic cartridge design can be combined with assay-specific reagent filling as a final step. Controlled reagent release is achieved through mechanical actuation. A plunger applies force to the blister dome, opening a frangible seal. Continued compression displaces the stored liquid. After an initial release pulse, the flow rate can be maintained at a constant level. This blister is also available as a standalone module, making it well suited for early testing with 3D printed parts or rapid prototyping cartridges. An alternative to blister storage is the use of integrated liquid tanks that are part of the molded cartridge backbone. This approach is often preferred when very large or very small volumes have to be stored, or when the number of required blisters would consume too much cartridge space. These containers are formed directly during the injection molding process, eliminating additional assembly steps. Each tank includes a fill port that is permanently sealed after filling, as well as valves on both ends. We typically implement these valves as frangible seals that can be opened mechanically by the instrument in a simple and robust way. Liquid displacement requires a driving force. This can be supplied pneumatically by the instrument or achieved through an on-chip pump using external mechanical actuation. Elastomeric pump bellows integrated into the cartridge enable a fully closed system and remove the need for pneumatic interfaces. Liquid tanks support complete reagent displacement, allowing volumes to be accurately pre-metered during filling. Let's now switch to dry reagent storage. Many point-of-care assays rely on dried or lyophilized reagents, particularly for more sensitive biological components. While liquids are often used for buffers, drying or lyophilization is key to achieving the required shelf life. Two standard approaches are commonly used: drying reagents directly in on-chip cavities, using lyophilized pellets or beads. Both methods are well established, but each presents challenges, especially in high-volume manufacturing. While pellets such as lyo beads offer advantages over on-chip drying, those benefits are only fully realized if the cartridge is designed with the proper integration interface. Reagents must be integrated at a manufacturing stage where downstream processes do not compromise their stability, and full biological and fluidic functionality must be maintained upon reconstitution. On the next slide, we'll introduce an advanced solution that simplifies this workflow. Using a carrier to integrate immobilized reagents into microfluidic devices is a powerful approach, as it decouples reagent immobilization from cartridge assembly. This separation allows both steps to be optimized independently. Carriers can be prepared in large batches, simplifying quality control, improving yield, and enabling verification at intermediate production steps. It also increases flexibility for using the same device platform across multiple applications. DINS developed the reagent plug as a carrier solution for dry reagents. It forms the basis of a workflow platform designed to support efficient, high-throughput reagent drying and robust handling during manufacturing. Let's walk through the workflow. Reagent plugs are injection molded independently of the cartridge and arranged in an 8 by 12 carrier plate. Reagent solutions are then dispensed onto the plug's immobilization surface, followed by drying or lyophilization. These final steps can also be performed at the customer site if desired. For assembly, the carrier plate is flipped upside down so a robotic arm can pick individual plugs and place them into designated ports in the cartridge. If required, the plug can be permanently sealed into the cart. Reagent plugs can be customized in both size and shape to accommodate different reagents and solution volumes, providing a high degree of flexibility within a standardized platform. As the reagent plug is manufactured independently of the backbone, the adsorption surface can be better tailored to match the requirements for the immobilization of the particular reagent. What if you want to use lyophilized pellets? The reagent plug can also be combined with lyo beads. When manufactured from a thermoplastic elastomer, the plug provides additional functional advantages. In this configuration, the pellet is first loaded into a chamber on the cartridge, which is then sealed with the reagent plug. The plug can be mechanically deflected, enabling actions such as pellet crushing to enhance mixing during reconstitution. After reconstitution, the plug can remain compressed, reducing dead volume in the chamber. That brings us to the end of today's webinar. Thank you for joining us for this session on the storage and handling of reagents on microfluidic cartridges. If you have questions or feedback, please feel free to reach out to our experts via email. We hope today's presentation provided practical insights, particularly around manufacturability and scalable design. Our goal is to deliver relevant, hands-on guidance that supports your day-to-day work. Thank you again for attending, and we look forward to continuing this journey with you in future webinars.